Okay, we're going to make a start now. So, welcome everyone to our A Level AQA Biology session. In today's lesson, we are looking at phagocytosis. Okay, so we've looked at cells, we've looked at cell membranes. Now we're going to move on to immunity. It's a long unit, but we'll move through it topic by topic. We're going to start off by looking at phagocytosis. So we're going to zoom into the structure, um, not the structure, the process of phagocytosis. And we are going to look at some mock schemes from questions that have been asked on phagocytosis. So exactly the points that you need to learn and what you need to write in an exam to gain all the marks. We're going to go through the process, and then we've just got a little bit on antigen presenting cells just to um, prepare you guys for the next stage of immunity. Okay, so we're going to start straight into it. So in terms of immunity, the first part of the spec focuses on basically your specific response and your non-specific response. Okay, today is your non-specific response, which is your phagocytosis. It's a build-up from GCSE, and it's just going to be going into a little more detail. There are key points and key things that you need to know for each of the processes in phagocytosis. Okay, so if we just skim over this spec as we're going through a new topic, you need to know about how each type of cell has a specific molecule that is embedded on it. So it's normally receptors, and those receptors, they're proteins, and they allow the cell to identify pathogens, cells from other organisms of the same species, abnormal body cells, and toxins. So that's the purpose behind your cells. Your cells have receptors which can detect pathogens. You need to know what an antigen is. It says over here clearly, definition of antigen. You need to know how to define antigen, the effect of antigen variability on a disease and disease prevention. We're not going to be looking at that today. I'm just giving you a bit of an overview. But basically, all that means is why do we have so many mutant strains of diseases? For example, if we look at COVID, we have many strains of COVID. And the reason why is because the antigen that comes from it keeps varying its shape, which means that our body, even after receiving immunity, even after being infected with the virus and our body producing antibodies, we aren't able to recognize that antigen from the COVID because it's sort of changing its structure. So our body thinks it's something else when in reality it's just another strain of COVID. You need to know phagocytosis of pathogens, the process of it, and the subsequent destruction of ingested pathogens by lysozymes. So that's part of the process which we're going to go through today. The next thing is the your cell mediated and humoral response. Your cell mediated response is including your T cells, and your humoral response is your B cells and your phagocytes. You just need to know the specific roles of your T cells and your B cells and your phagocytes. Um, B lymphocytes, again, we're not going to look at in today's lesson. This is part of your specific response. Your phagocytosis is non-specific because any pathogen which comes into your body, your phagocytes, which is a macrophage, they will go and attack that pathogen straight away, no matter what pathogen it is. It's not involved in really creating sort of antibodies, in creating memory cells. That's what your B cells do. Okay, and your T cells can turn into different types of cells. They can become into a T helper cell. They can become into a cytotoxic T cell and kill the pathogen. But phagocytes, they are just exposing the antigens, so the immune system is aware that a pathogen has entered your body and they can produce antibodies against that pathogen. You need to know the definition for an antibody. You need to know the structure of an antibody. You need to know how an antigen antibody complex forms. And again, that's not in today's lesson. I'm just going through the bullet points that you need to know if you've already studied this topic beforehand. You also need to know the role of plasma cells and memory cells. Okay, and vaccines, there's a bit on vaccines which gets asked in sort of application questions in biology papers. You need to know the concept of herd immunity, how to define it, and you need to know the difference between active and passive immunity. Okay, so there's another part of the spec where you look at monoclonal antibodies, but we're going to focus on this part first, and we're going to split it maybe across 3 lessons so we can get through all the points on the spec. So, moving on, so just a bit of an introduction to immunity. Okay, now we know that in our body is very well adapted, and it has specific chemical and physical barriers that you probably studied at GCSE. For example, skin is a physical barrier. Okay, the skin protects all your blood, it protects everything which is inside your body from getting infected from any pathogens. So it's a physical barrier. Okay, um, you've got hairs which trap the pathogens to prevent them getting in sort of from your nose, through your ears or anything like that. That's another example of a physical barrier. In terms of chemical barriers, you've got your stomach acid. That's your main chemical barrier. So if a pathogen does get through all the way to your stomach, if you've ingested a pathogen accidentally through a surface, then it goes down to your so through your esophagus and into the stomach, and your stomach acid will destroy the pathogen there. Now, if a pathogen gets past all those forms of your physical and chemical barriers, then it will enter the blood. Okay, and if it enters the blood, then it will trigger an immune response. So that's basically what's happening. You have your chemical and physical barriers to start off with, and they will try defend your body as best as they can. However, if those chemical and physical barriers fail, which they sometimes will, that's why you feel ill, the pathogen would have got into your bloodstream. And if it's in your bloodstream, that will trigger an immune response, which is led by your white blood cells. And that's your second line of defense. Your white blood cells are your second line of defense. Your um, first line of defense are your chemical and your physical barriers, which will hopefully prevent the pathogen from entering your body. And there are two types of responses that white blood cells can trigger. They can either trigger a specific response or they can trigger a non-specific response. And today, what we're going to look at is the non-specific response, which is your phagocytes. Okay, your lymphocytes, your B cells, and your T cells, those are your specific responses. Okay, lymphocytes are just an example of a white blood cell. But phagocytes are your non-specific response. Okay, so they don't respond to certain shapes or receptors. They literally are attracted to chemicals which they see as foreign. Okay, and what they will do is they will engulf that chemical. You would have probably heard of that at GCSE. They engulf that chemical and then they present some of the proteins and becomes an antigen presenting cell, which triggers the lymphocytes to sort of produce your antibodies within your body. So, phagocytes. Now, again, as I said, you've come across phagocytosis at GCSE, but you probably went over the really basics. So, engulfs produces antibodies and produces antitoxins to deter the pathogen. Now, a phagocyte is a type of macrophage. Okay, it's a macrophage, which is just a type of white blood cell, and the phagocyte is what actually carries out phagocytosis. And they are found all around your blood and your tissues. Okay, they're just circulating your blood in case that they come across anything that shouldn't be in your body, i.e., they come across a pathogen. And phagocytosis, as I mentioned, is a non-specific response, which means that any non-self cell detected by the white blood cells, they will cause the phagocytes to trigger the same response and destroy it. Okay, and this is why when you have sort of an organ transplant, you have to have the same DNA as the person donating the organ, or there is a risk that your organ may fail. The reason being is because your immune system will detect the DNA as something foreign, something that doesn't belong in your body. It will treat it as it would do to a normal pathogen and it will go and attack that pathogen. And if it does go and attack that pathogen, then it will destroy that organ and that organ will no longer have its optimal function, which means that you will end up dying because of your immune response. So again, phagocytosis is part of your non-specific response. Any non-self cell which is detected by the white blood cells will trigger the same response. Okay, even if it's helpful or if it's not helpful, if it's a non-self cell and it doesn't belong in your body, or it's something foreign that the body doesn't recognize, it will trigger the same response and it will go and destroy it. Okay, so that's what phagocytes are. They're a type of macrophage found all around your body, in your bloodstream. They are part of the non-specific response, which means anything foreign that they detect, they will go and attack it, and they will cascade and trigger an immune response to get rid of it and produce your antibodies against it. So this over here is the whole process of phagocytosis. Okay, and we're going to go through a method. First of all, we're going to go through a longer method, and then we're going to condense that method by looking at a mock theme of what mock theme actually wants to see when you get asked a question on phagocytosis. So this we're going to be referring to this image again and again whilst we're explaining the process. So we will come back to this process, this image. So the process. So, as I said, phagocytes, they're just circling your body. They are present in your blood. They're just going around your body, trying to detect any chemicals or any pathogens, or any debris that shouldn't belong in your body. Okay. Now, what happens is when something doesn't belong in your body, okay, chemicals are released from that thing. Okay, so if it's a pathogen which has entered your body, that pathogen will release certain chemicals. Okay, it will release certain chemicals, and these phagocytes, they will detect that release in chemical. Okay, and they will be attracted towards the chemicals or towards the abnormal cells. Okay, because that's the thing with phagocytes. The reason why they are adapted for their function is that they have the ability to detect these chemicals which are coming from these pathogens. And pathogens will release different types of chemicals, but it doesn't care what type of chemicals the pathogen is releasing. If something is releasing chemicals or something abnormal is being released, these phagocytes will be attracted towards them. Okay, so pathogen has entered your body. Your pathogen has started releasing chemicals within your body, and that's attracted the phagocyte towards the pathogen. Now, the phagocyte has many receptor binding points. Okay, so on a cell, if I draw a cell of a phagocyte, so we've got a phagocyte over here, there are many sort of receptors which are present on the cell, like that. Okay, and all around. There are many of these receptors. And what will happen is that these receptors, they have so many of these receptors that any chemical or antigen, we're going to come on to the definition for antigen, but basically pathogens, they have a specific antigen that is present on their surface as well. Okay, and they have the ability to bind to the receptors on this phagocyte. Okay, so on this phagocyte, as I mentioned, there are many receptors, and these receptors, they will bind to chemicals or antigens. Antigens are just proteins. We're going to go through the definitions, but antigens are just proteins. Okay, and they have so many phagocytes have so many receptors on their surface that they will be attracted to they will have the ability to bind to these proteins, these antigens, these chemicals that have been released by the pathogens. Okay, so pathogen has entered the body. It started releasing chemicals and proteins in your body. The phagocytes which are circling your body have detected those chemicals being released. They are going to be attracted to the chemicals that are being released, and they're going to go, and because they have so many receptors on their surface, the complementary receptor is going to bind to the complementary antigen or the complementary chemical which is being released. Okay, and once it's bound, the phagocyte is going to change shape, move around, and engulf that pathogen. Okay, so engulfing is the same as GCSE. So at the moment, what's happened, we've just gone into a little bit more detail compared to GCSE. In GCSE, the first step was engulf. Yeah, white blood cell phagocyte engulfs pathogen. Over here, what you need to know is that chemicals or antigens, they are released by the pathogen once they have entered your body. Your phagocytes have the ability to detect the chemicals and the antigens, and they are attracted to those chemicals and antigens. Phagocytes have many receptor binding points, which means that they have the ability to bind with the antigens or the chemicals that have been released by the pathogens. Now, once they have bound to these antigens or these chemicals, the phagocyte will then change shape, it will move around the pathogen, and it will engulf it. Okay, it will take the pathogen in. So if we just go back to our image that we've been looking at, we've looked at these first two steps over here. So we've got a bacteria. Okay, this bacteria is the pathogen which is causing the disease. Okay, so we have our bacteria. Now the bacteria is going to have is going to release certain chemicals, it's going to have certain antigens. And these are your antigens from the bacteria. Okay, so these circles are your antigens. So these are your antigens. Okay, and remember I said the antigens are just proteins. And what's going to happen is that this phagocyte, this big phagocyte, has been attracted to the bacterium which has just entered the body because of the antigens and because of the chemicals that it's releasing. And it's moved towards the bacterium. And what it's going to do is, because it has, remember, phagocytes have lots of receptors. So these are examples of receptors on the phagocyte. Okay, so these are your receptors on the phagocyte. And these receptors, they are going to bind to the antigens that are present on the bacterium. Okay, so as you can see, this one over here has bound. And once it's bound, the receptor has been activated, which means that the phagocyte can take in the pathogen. So what happens is that this phagocyte will take in the pathogen, it will surround the bacterium, and it will engulf it. Okay, you need to use that keyword. Pathogen is engulfed. So that's what's going to happen first. Now, the next couple of steps we're going to look at. So we've looked at the first three steps. The next step is a keyword. Okay, so once they've engulfed the pathogen is inside the phagocyte. The pathogen is contained within a part of the phagocyte. It's contained within a part which is called phagosome vesicle. Okay, so it's not going to take up the whole phagocyte. It's not going to be just moving around randomly in the phagocyte. It's going to be contained within like a barrier. It's going to be, if we think about a circle in the phagocyte, it's going to be contained within a sort of a capsule within the phagocyte. And you call that phagosome vesicle. Okay, so make sure that you learn that name. That's really important for exams. Okay, so if we go back over here to this image, over here, we had an open bacteria that was entering the phagocyte. Nothing was happening to the bacteria, it was just entering the phagocyte. And as you can see, after it's been engulfed, there's this capsule which is around it. You can see that this capsule is contained within this circle. And because it's contained within that circle, this circle is called a phagosome vesicle. Okay. Okay, so once engulfed, it's contained within a phagosome vesicle. And then what happens is a lysosome, which is a key part of phagocytosis. And we looked at that during our cell structure. One of the functions of lysosomes is to take part in phagocytosis. Phagocytes, they have lysosomes which are present within them. And what will happen is that the lysosome that is present within that phagocyte, it will fuse with the phagosome vesicle and release its contents. Okay, so the lysosome has an enzyme. Okay, and that enzyme is called the lysozyme enzyme. Okay, so let's just look at what's going on. Okay, so we have this vesicle. This is our phagosome vesicle. And in our phagosome vesicle, we have our bacteria. Okay, so we have our bacteria in our phagosome vesicle. What's going to happen is that a lysosome is going to come across. Okay, so these lysosomes, they're going to move towards this, and they're going to fuse with the phagosome vesicle. Okay, and inside these lysosomes, you're going to have an enzyme, which is the lysozyme enzyme. So inside lysosomes, you have an enzyme, which is your lysozyme enzyme. Okay, and that lysozyme enzyme enters the bacteria, it enters this phagosome vesicle. Okay, and lysozyme enzymes, you need that, and again for exam, it's a lytic enzyme. You don't need to know that, but the purpose of the lysosome enzyme is to hydrolyze the pathogen, to break down the pathogen. That's the purpose of the lysozyme enzyme. So what's going to happen at the moment, in step 4, we had the bacteria being contained within a phagosome vesicle. Okay, that phagosome vesicle is going to fuse with a lysosome, and that lysosome has a lysozyme enzyme within it. Okay, that lysozyme enzyme is going to enter the phagosome vesicle where the bacteria is contained within. And the lysozyme enzyme, it's going to hydrolyze the pathogen. It's going to break down the pathogen. Okay, which means that the pathogen is destroyed and the soluble products are absorbed and used by the phagocyte. So even in a pathogen, there are useful products that the phagocyte can use. So the useful products that it has, it can just absorb those products and use it for itself. Okay, so that's the process of phagocytosis. I'm just going to go back to the image and cover the final few steps. So at the moment, we had a bacteria. Okay, bacteria had antigens on them, which are just proteins. They are going to be releasing certain chemicals that are going to be attracting phagocytes towards them. Phagocytes have many receptors on them, and the complementary receptor is going to bind with the antigen present on the bacteria surface, and that's going to cause the phagocyte to wrap around the bacteria and engulf the bacteria. Once it's been engulfed, the bacteria is contained within a phagosome vesicle. So this circle around the bacteria, it's contained within a vesicle. And what happens next is that lysosomes, they towards the vesicle and they fuse with the vesicle. Okay, so as you can see, these green structures are your lysosomes and these black structures are your lysozymes, which is just an enzyme that hydrolyzes the pathogen. So the lysosomes, they move towards the phagosome vesicle. So they move towards, and as you can see, they are fusing. Okay, so they are fusing over here. And when they fuse, all the lysozyme enzymes that are present within the lysosome, they are released into this phagosome vesicle. And once the lysosome enzymes enter the phagosome vesicle, they will digest the bacterium. They will hydrolyze the bacterium. Make sure you use hydrolyze. Okay, they will hydrolyze the bacterium. And all the waste, the waste that is sort of produced is just released by the self. by the cell, and the soluble products, they are absorbed and used by the cells. Okay, and that's the process of phagocytosis that you need to know. I have gone into a little bit more detail. The only reason is so you guys get a better understanding of what's actually happening. In an exam, it's going to be worth around 3 marks. And I've picked out 2 exam questions which are asking for the same thing. One is not an application question and one is an application question. However, the mark scheme is bang on the same for both of them. Okay, so we've gone through the process of phagocytosis. The last thing before we move on to exam questions, we're going to look at antigens. Okay, now antigens, you need to know the definition for antigens. We looked at that on the specification. It says that you need to know the definition for antigen. So basically, what can happen when a phagocyte has engulfed a pathogen and when it's broken the pathogen down, some of the antigens can be released into the phagocyte. Okay, because remember, some antigens are present on the outer surface of the pathogen. Once that pathogen has been broken down, the antigens are present within the phagocyte. So what can happen is that the phagocyte can become an antigen presenting cell to trigger the next part of the immune response. So remember, we need to create antibodies. We need to create memory cells. In order for that to happen, a phagocyte can become an antigen presenting cell. And an antigen presenting cell, all this will do is it will present the antigens on the surface. And these antigens, um, they will activate sort of B cells to produce um, antibodies. Okay, so remember, we need antibodies, and in order to know what antibodies we need the antigen as well. So it becomes an antigen presenting cell, so the next part of the immune response can be triggered. So antibodies or memory cells can be produced. Okay. And the definition for an antigen is a foreign protein which stimulates an immune response. That's a 2 mark definition there. This is your first mark, foreign protein which stimulates an immune response. That's your second mark. Okay, so without antigens, antibodies can't really be made.